Hey there, I'm going to go over the lab for lesson 2.02, limiting factors. In this lab, I'm going to show you everything you need to do and stick with me through it, even if this is long. Go ahead and pause the video and do that portion of the lab on your own, and then come back to the video and continue working slowly throughout all of the steps that I show you so that you can get the best score possible. I'm going to recommend also using a split screen if you know how to do that. If you don't know how to use a split screen on your device, go ahead and YouTube how to split your screen and you'll find out different ways to do that. Because you can see right now I have 3 tabs open, and I will definitely be working heavily between 2 of these tabs in order to complete the lab. One tab, well, 2 tabs right now are open to Buzz. One is open to the course card. The other is open to the last page of the assignments. And then finally, I have a copy of that lab report open and ready to go too. So when you come to the course card and you scroll down for 2.02, you're going to see that I have created, once again, a Google Doc copy. Now, if you are a fan of Google Docs, or if your system will allow you to make a copy, that's great. If you don't want to use Google Doc, or your Chromebook or whatever device that has been issued to you doesn't allow you access to the Google Docs I share, you're going to want to use the lab report on the last page of the lesson. So that lab report would be right here. I am on page 6 of 6 in the limiting factors lesson 2.02, and the actual report is right here. The grading rubric that I use to grade you is also located right there, so you can see exactly what you need to include in your lab report to be a skilled scientist and get all of the points. All right. In this lab, you will explore how limiting factors affect a native population of cricket frogs in the Everglades ecosystem. You will learn more about the relationships between cricket frogs, food source, pollution, and predators. Objectives: identify various limiting factors, both abiotic and biotic, in an ecosystem. Describe the impact of limiting factors on native populations. Explain how the various limiting factors may influence each other, for example, parasitism and the spread of disease. And predict the long-term effects of limiting factors on a local ecosystem. All right, if you've forgotten what limiting factors are, they told us up here. The relationships between cricket frogs, food source, pollution, and predators. One of the things you need to do in this lab report is put the objective. What is the reason you are doing this? Right here at the top, first you're going to put your name and title of the lab, and then next, in your own words, what is the purpose of this lab? That is something that I've just gone over. So if you're confused about why you are doing this lab or what the outcome is supposed to be, make sure you take a close look at this page. All right, here are some instructions on how to use the simulator, and then they're going to welcome us to the Everglades. The Everglades is a complex and varied ecosystem or environment located in South Florida. It is made up of hardwood hammocks, mangroves, pinelands, lowlands, freshwater, marl prairie, cypress, and marine environments. It supports plant and animal species found nowhere else on Earth. There are many limiting factors that help determine the size and distribution of populations. These factors can be organized into 2 categories: abiotic, or non-living, and biotic, or living. Some examples of abiotic factors or non-living factors include temperature, extreme weather, and sunlight. Biotic or living factors might be birds, disease, and mammals. Cricket frogs are a native species in the Everglades. They are a species of tree frog with rough, warty skin in a variety of colors. Some are black, brown, red, green, or gray, but all have a bright stripe of color running from the tip of the snout and down their backs, broken by a triangle pattern between the eyes. Here's a fun fact: cricket frogs are not named for their cricket-like jumping abilities. They take their name from the cricket-like call they use to attract mates and keep away other mates. Here are a few limiting factors that directly affect the cricket frog population in the Everglades. Select each factor to learn more. Predator: alligators. are a common predator found in the Everglades. They are primarily found in freshwater swamps and marshes, but are also found in rivers, lakes, ponds, and mangrove areas. They feed on fish, amphibians, snails, crustaceans, snakes, and mammals. Pollution. Pollution is the introduction of any substance or item that is harmful to the ecosystem. In the Everglades, this includes garbage and agricultural runoff from fertilizers and pesticides. These pollutants disrupt the delicate balance of the ecosystem and may cause native organisms to die off. Food source. Mosquitoes provide a food source for many organisms that live in the Everglades. They can be so numerous in this unusually wet place. Sometimes people have to wear bug suits to protect themselves just to walk a few steps inside the Everglades. Mosquitoes also carry and spread diseases such as West Nile virus and malaria as they move from organism to organism, sucking blood. All right, hypothesis. So now we are on this next section here. In this section, you're going to include the if-then statements that you developed in the lab activity. They are copied here for you, and you will need to make your predictions. If the predators of the cricket frog are increased, the cricket frog population will. I'm going to choose stay the same for all three. You can choose whatever you think the best answer would be. If pollution is increased, then the cricket frog population will, over the span of 5 years. And if the amount of food available is increased, then the cricket frog population will, over the span of 5 years. Again, you pick yours. And right here it says, be sure to record your hypothesis in your lab report. So you can come back here and you can delete the words that wouldn't belong to make that statement true. You can rewrite the statement however it works for you. Then we move on to the procedure. To begin your investigation, let's start with the predator limiting factor. You will observe how increasing or decreasing the predator, alligator, impacts the population of cricket frogs in the Everglade ecosystem over 5 years. So we are predicting, we're going to see what happens initially, and we're making predictions 5 years from now in this lab. So I'm going to go ahead and open this. The alligator in this simulation, you will examine how alligators affect the cricket frog population. Count how many frogs are on screen before beginning your trials. Be sure to make note of it so you can record the data in your data chart after the first simulation. Then you will run this simulation twice, adjusting the slide for a trial at low, which decreases the amount of predators or alligators, and a trial at high, which increases the amount of predators. Select begin to start your investigation. All right, so we are now in the simulator. At the bottom here, you can see the frogs, and you can count them for an initial count. Over here, we have a key. One frog equals 5,000 cricket frogs. So if I count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, there are 10 frogs in the model. That means 5,000 times 10 is 50,000 to start. So I'm going to come into my lab report. Oh, I'm going to need to talk about this, so I'll come back. And my beginning frog count, I would enter 50,000 for predators. All right, now I'd come back and I would select the low. And I want you to watch what happens to the graph because you're going to be describing that. So I'm going to go and see what happens. Oh, it's going up and up and up. So if the predator is low, our cricket frog population increases. So our beginning count was 50,000. And simulation 1, where did it end in 5 years? It ended at 90,000. I'm going to save that and close. Then I'm going to adjust and I'm going to say, what happens if we have a high predator count? What if there are a lot of alligators? Let's click play and watch what happens. Oh, it's decreasing. Oh, wow, it's still decreasing. Wow, that is a huge decrease. It went from 50,000 and ended at about 15,000. Save that and close. Then we can go to the next limiting factor, which is pollution. In this simulation, you will examine how pollution affects the cricket frog population. Again, count how many frogs are on screen before beginning your trials. Be sure to make a note of it so you can record the data in your data chart after the first simulation. Then you will run this simulation twice, adjusting the slide for a trial at low, a small amount of pollution for 1 year, and a trial at high, a large amount of pollution for 2 years. Select Begin to Start. So if we count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, same as last time. Adjust for low pollution, run the test, record your results, run for high pollution test, and record your results. All right, I have finished collecting my data. Make sure you're watching the graph line that you can describe what happened. Finally, we're going to look at the food source, mosquitoes. In this simulation, you will examine how the amount of mosquitoes affects the cricket frog population. Count how many frogs are on the screen before beginning your trials. Be sure to make a note of it so you can record the data in your data chart after the first simulation. Then you will run this simulation twice, adjusting the slide for a trial at low, which decreases the amount of food, and a trial at high, which increases the amount of food. Select Begin to start your investigation. Okay, so once again, you're going to count for your beginning number of cricket frogs to use as a comparison after you adjust the limiting factors. You change the amount of food that's available to see and count or measure how many cricket frogs are there with low food availability or high food availability. All right, once you complete all of your trials, you're going to see that I have not entered incorrect information here because you need to get your own results. So do not copy the numbers or letters that I have in the boxes. They are not accurate. But once you have your chart completed, you're going to take those numbers and copy and paste them into this data chart right here. Then I'm going to bring you back up to the top, to the procedure. The procedures are listed in your virtual lab. You do not need to repeat them here, but you do need to identify the test variable, which is the independent variable, the outcome variable, which is the dependent variable, and the control group for this experiment. Remember, the test variable is the item that is changing. What did you change? You slid the lever back and forth. What were those things called? The outcome variable is the item that you are measuring or counting. And then the control group is the group in the experiment where factors being tested are not applied. So before applying those limiting factors, the control group, what did we measure? It's like a baseline that we can use to compare. So make sure you list those 3 things here. This is often the part of a lab that students are forgetting to complete. All right, so you're almost done. Now you are going to do some description. You're going to use the graphs either that you printed or you observed in the simulation. Those lines, what happened when you had the limiting factor of low alligator population? What happened to the red line of cricket frog population? What were the trends that you noticed? And when you put pollution as a limiting factor, what were the trends you noticed with low pollution versus high pollution? What happened in that graph? And finally, for the mosquito food population. If mosquitoes were limited and there was a low supply, what happened to the cricket frog population? What did you notice on the graph over the course of 1 year, 2 years, 3 years, 4 years, and 5 years? And then finally, you're going to answer these 6. Make sure all 6 questions appear. I've noticed some lab reports where students decide they're going to eliminate a question. There are 6 on the original document. And also, please write incomplete sentences. Answer all 6 questions, and you are ready to turn in your lab report for 2.02. If you have questions or want extra help, book an appointment with me. You can use the booking appointments right here on the landing page, or you can send me an email, and you can send an email by clicking right here. Bye, guys.